Hello, student. We will continue with the lecture. That is, the next part is randomized block design. That is RB design. So, in this particular design, it is again the improvement over CR design. That is which we have seen in the last lecture. Complete randomized design. So in RB design, its a principle is the third point, that is the local control. And with the local control, all other two principles, that is randomization and replication, all these principles are applied for this particular design. So in the RB design is in RB design, the subjects are first divided into groups, which you can say blocks, and those groups, within those groups, the subjects are relatively homogeneous with respect to some selected variable. Now, what it means that subjects or experimental units which are grouped into a blocks, which are called as blocks, with the different treatments to be tested randomly and assigned to the units of each block. So, essentially, the randomized block design group subjects that share the same characteristics together into blocks and randomly test the effect of each treatment on individual subject within the block. I know you couldn't get anything. Now, with the example, we will try to understand it. Now, suppose in a farm, we want to test the plant disease, right? In a farm, a particular corn plant is there and it is getting affected with a plant disease. That plant disease is caused by a fungi. Now, the plants are getting widely affected because of this fungal, because of this fungi, and we want to find out efficacy of different fungicides. So, against the which fungicide will be better to treat this particular fungal disease, we want to test that. So for that, what you can do under this particular design, we want to see which fungicide is controlling it, right? So you can split the field into blocks and randomly treat the section of each block with various function which could exist in the field. In a sense, you have to suppose you are having a 10-acre field, right? 10-acre field, divide that field into 2 acres each, right? And now equal division should be there, right? Which you treat it as a block. So if you are divided it into 2, it means total 5 blocks will be there, right? Now, those blocks you are taking it randomly. You are just randomly selecting one corner, then and just divided it into the 5 parts, which each part is of 2 acres, right? Now, uh, when you do this and give the treatment of different fungicides to each block, right? Now, when you're doing this, what will happen? One section of the field may have more shade, right? Another will have the extended wetness. Another will have a very good sunlight, right? Another area may have the ideal environment for the pathogen to proliferate, that is the wetness. Wetland is there, which will remain wet because the sunlight is not reaching to that particular area. Now, another section may have a slightly different type of slope. So just consider that out of this 5 sections, one section is having a lot of wetness, section 1, or block 1 you can say. Block 2 is having a lot of sunlight. Block 3 is having some different type of soil, right? So soil fertility will be the another factor. Then fourth will be having again the wetness and very shady area it is, where the pathogen, that is the fungal, can proliferate more. Another, the fifth one may have a lot of slope, right? So now through blocking, when we do the blocking, the farm can now excuse for these potential confusing factors. Now, all others are the confusing factors, right? And you are treating all 5 sections with the same fungicide, for example. So, Roundup, whenever you are making all this collectively, you are going to study and finding out the effect. Overall, if you do the summarization, it will be having excuse, in the sense you are leaving all these factors. You are not focusing on all these factors. Why? Because you have randomly done the block design. So generally, this type of design is used in the studies of agriculture sciences or animal sciences or pharmaceutical studies. So generally, this RB design is that in which each treatment appear the same number of times in each block. So if you have given a concentration of fungicide that is 5 ppm, so in each block you have to give 5 ppm concentration of fungicide only. So the RB design is analyzed generally in two-way analysis of variance, that is two-way ANOVA. Now there are often confounding variables, that is confusing variables, which affect whose effect cannot be predicted. So you cannot predict the effect. There are some such factors of which in generally in case of pharmaceutical or in case of animal sciences or agriculture, you plan the experiment, but some uh, unpredicted variables are there. Like even though there is a summer, then rain is coming. So that is unpredictable. You cannot predict it. So a researcher might oblivious to even the existence of confounding variables. So in such a case, understanding a project and being able to predict potential sources of variation is very important for getting accurate relationship between the dependent and independent variables. So this is the design, this particular design, randomized block design, gives benefit of uh, benefit of all these factors, and which is actually beneficial for this particular type of design or this particular type of experiments, I mean. Now, the next one is Latin square design, where the experimental design very frequently, this is again the experimental design which is very frequently used in agriculture research, and mostly, why? Because in agriculture, uh, investigations which are carried out mostly uh, mm, in a different way as compared to all other studies. Why? Because there are some natural players, in the sense rain is there, or sunlight is more sunlight, summer is there. You cannot just predict. Sometimes it happens opposite. So, or some you can say that sometimes wind flow very I mean storm is there, or that is unpredictable, that you are not predicting when you are planning your research plan, right? So in such situation, this Latin square design is generally useful. So for example, we can take the example that you want to see the effect of 5 varieties of fertilizers on the yield of certain crop. You can say V2 crop you want to judge. So there are 5 fertilizers, crop is one, right? Now, in case of varying fertility of soil in different ways, so you just again take the field, divide it into 5 blocks, right? And five blocks, 5 treatments you are going to give, right? So divide it into that. But in each block, there might be different fertility, because fertility may changes from distance to distance, from soil to soil. So it may happen that in some area of block, a very good fertility of soil is there, and in some the soil is not more fertile. So you can consider that. And another factor is varying seed on the yield. So there are we generally buy the seeds by seeing the proper condition of the seed, but still there are some seeds which may not get fertile. So there are varying types of seeds, and as the seeds are getting vary, it will again uh, affect on the yield. So there may be the impact of varying seeds on the yield also. So there are two extraneous factors are there. One is soil fertility and another is varying seed. So whenever LS design is used, generally, whenever there are two major external factors are there. So in this case, soil fertility and seed differences is the extraneous factor. And what you want to find it out, that treatment of different fertilizer. So these are the 5 levels of the fertility of the soil. These are the seeds differences, 5 seed differences they have taken, and A, B, C, D, and E are the different 5 fertilizers. So if you can see that in each column, all the all the fertilizers are there, A, B, C, D. If you take the row, all the fertilizers are there, even though they are not arranged exactly in the same way. They are randomly arranged, but still all fertilizer treatment is given in all rows and in all columns. So generally the LS design is very similar to the two-way ANOVA technique, and the differences in the fertility gradients in the field to be eliminated in comparison to the effect of different varieties of fertilizers on the field of the crop. And even the seed, this particular design, this is the merit of the experimental design, and the limitation of this design is, it is that although each row and column represents equally all fertilizer varieties, everywhere the fertilizer is getting covered in that particular row and column, still there may be considerable difference in row and column. Means both up and across the field can be there. So you can remove that also by taking means of the rows and column equal to the field mean, and by adjusting the result. So you can still go to the accuracy level just by taking means of rows and columns to the field means. Now, another limitation of this design is it requires number of rows, column, and treatment to be equal. Everything should be equal. If it is unequal, in case 2 into 2, only 2 factors you are taking, rows and column, or column and treatment you are taking only, then there will be no degree of freedom available. And thus this design won't be able to work good, efficiently. Then if the treatments you are taking more than 10 or 10, then also the size will be larger, and then the columns and rows may not be homologous, right? And thus again, it will not be able to work effectively. So therefore, generally LS design of orders 5 into 5 to 9 into 9 are used. Now the next is factorial design. This is one of the best design, you can say, which is used to do the research. So factorial designs are used in the experiments where the effect of varying more than one factor are to be determined. Now there are types of the factorial design which we are going to see, but this is very important design which is very economic, and it is also having social phenomena where usually the large number of factors affect in a particular problem. So factorial designs can be of two types. One is simple factorial design and another is complex factorial design. So we will see one by one. Simple factorial design, in which generally we consider effects of varying two factors on the dependent variable. So whenever only two factors are considered, it is called as two factor factorial design, whereas in complex design more than two factors are there, so it is called as multi-factor factorial design. So simple factorial design can be 2 into 2, that is simple one, or it may be 3 into 4 or 5 into 3. So we can see one example here, that this is the example of simple factorial design, where for example, in this particular design, any number of independent variable with any number of levels you can take. So you can take any number of levels and any number of independent variable. So, for example, an experiment could include, suppose one experiment, a researcher is doing experiment on a group of people. It want to give some psychotherapy. So what type of psychotherapy it is going to give? It is going to give cognitive versus behavioral. Cognitive means mental psychotherapy, mental therapy, and behavioral means the communication or how the person behaves. That all therapy that's researcher want to give to group of the people. Now the length of the psychotherapy, here two types of psychotherapy, that is cognitive and behavioral. Length of the psychotherapy is again 2 weeks versus 2 months. So from 2 weeks to 2 months it want to give. And again the psychotherapist is going to use the sex, that is female and male. So for again here is two parameters. So this factorial design will be 2 into 2 into 2. So you can just see cognitive, behavioral. Therapist is going to give therapy to females as well as males. So you have to keep it the same number of female and males, and 2 weeks to 2 months. Thus it is 2 into 2 into 2. Now the next is complex factorial design, where this particular design which definitely have more than the two factors should get considered. So generally it consider 3 or more independent variables simultaneously, is called as complex factorial design. So in this case we are going to see the 3 factors with one experimental variable having 2 treatments. So 3 factors are there, 2 treatments are there, 2 control variables are there, and each one of which having 2 levels. Again 2 levels are there. So you can term in simply 2 into 2 into 2 complex factorial design. Now why 2 into 2 into 2? So there are 2 treatments, treatment A, treatment B. There are 2 levels, level A, level B, and there are 2 control variables, right, with level 1 and level 2. So in treatment A, there will be control variable level 1, control variable level 2. In treatment B also control variable level 1, control variable level 2. And in control variable 1, this is control variable 2. In control variable 1 again, level 1 and level 2. And these are the 8 cells or the groups you can say where the treatment can be studied. So all over there will be 8 cells where the treatment will be studied. Treatment A, treatment B, and control, that is level 1 and level 2. And everywhere there is a 2 levels. So the factorial designs are mainly used because of 2 advantage, because it provides utmost accuracy, it requires less labor, and thus it is gives the economically it provides the economy, and it also you can determine the main effect of 2 or more factors in one single experiment. So you have to do only one experiment, and in that only you can study 2 or more factors. So thus it is, because of that only it becomes economic. Then it also permits the comparison of interest. So you can compare the factors there itself, and the effect of such cannot be obtained by treating one single factor at a time. So by studying one single factor at a time and studying 2 or more factor at a time and their effect and their comparison is going to give you more data, more accurate data, as compared to studying only one single factor. So that's why this particular factorial design is always having a lot of importance. The determination of interaction effect is possible in case of factorial design. So interactive effects you can study well in factorial design. Now once you do, now you know that how many research plans are there, how many experimental designs are there, then you can actually develop a research plan. So whenever you want to do the research, you have to first prepare a research plan. And whenever you are preparing a research plan, what or how it is beneficial, why you should go for it, because it will organize your idea in a correct way. It will give you whatever plan you prepare, it will give you idea about flaws and inadequacies, if any, in your plan. It also provides inventory of what must be done, so you know that this has to be done, right? Which material you need to collect, what information you need to collect. You know all the steps very well. And then it it is actually the document or a plan or a outline which you should discuss with the experts or others and take their comment, critical comment, so that you can improve in that more and more, right? Then every research plan must contain following items. What? The research objective should be clearly stated. So whatever your plan is there in that, whatever objectives you are adding, they must be very clear, crystal clear, right? Then the problem which you are going to study, that also you have to clearly state that this is the problem and I am going to find out solution for this particular problem, right? Then the major concept should be very clearly stated. And the most important, that whatever concept you are tailing, it must be workable. Means haphazardly you are tailing such a solution that you cannot just work it out on, that is not acceptable. So whatever solution you are giving, it is workable solution you are going. Then in your research plan you must add the methods, which methods you are going to work on, which methods you are going to apply, and how you are going to solve the particular problem. You can give assumptions that if I apply this method, it may solve the problem in this way. So that concerning method you must clearly state in your research problem. Then you have to tell the detailed techniques which you are going to use in your problem. Then for example, if you are going to take an interview or you are going to use the interview method, then what type of interview, what nature of that interview will be there, what procedure you will follow while taking interview. Or if you are going to do the research by taking tests, then which conditions you are going to use when you are going to test these particular, or you are going to take these tests. What will be the instrument, nature of the instrument which you are going to follow for a particular test. Then if you are going to take the public records, then you need to consult it with the sources of the data. Then you should be the fact should be recorded in the research plan, that which type of public data, public records you are going to use, that should be included right there in your research plan. Then procedures for the quantifying data should also be written, that how you are going to quantify the data, that should be given very clearly, right? Then there should be, if you are going to use a population study or the sampling study, it should be very clearly mentioned what kind of sample you are getting, how you are going to collect the sample, what type of population you are taking as a sample from, the whether it is randomized, whether it is selective, that all you should mention it very well. How the sample is to be identified, to which method you are going to use, or which characteristics you are going to follow to identify the particular sample, and why you are taking that particular sample, how it is important for your problem to take that particular sample, that all should be mentioned in your plan. And which method you are going to follow to identify the sample, that all must be included in your research plan. Then the methods to be used in the processing the data. So once you do everything, then you may get the result in this way. And to analyze that research, which method you are going to use, are you going to use statistical method, or you are going to use some another method, indicate that clearly into your plan, right? Because that is going to save lot of your time and effort, because when the expert are seeing the plan, they will very clearly get the idea that if you are not using statistical method, they will immediately suggest you, no, you need to use here statistical method, because that will tell you the significance of your research. If you don't need to use and you are using, then they will tell, no, don't use statistical method here, you need to apply this type of software, this type of methods, better, it will give the better result. So then the results of pilot test, if you have done some research in that particular area, and if you have some pilot test results with you, that you must report in your research plan, because that will give the idea to the expert in the field that yes, how much you are whatever you are suggesting, the solution will get succeeded. And that will also uh, impact a lot, because if you have done the primary work, you are getting positive result, it means it will definitely give a good solution for whatever problem you are stating and you are trying to solve it. So and time and cost budget also, the researcher must always keep it into the mind and accordingly prepare its plan. Now we will see the video where the types of experimental research are explained well.
Hello students, today we are going to discuss types of experimental research part 2. Causal or experimental research is of two types, informal experimental design and formal experimental design. In the previous video, we discussed types of informal experimental design. In this video, we are going to discuss the types of formal experimental design. Formal experimental design is of four types: completely randomized design, randomized block design, Latin square design, and factorial design. We will discuss these types in detail. The first type is completely randomized design. Completely randomized design is also known as the single factor design and is useful in investigating the effect of one independent variable on the dependent variable. The independent variable will have a number of categories and each category is called a treatment level. In this design, the analysis of variance is performed to determine whether these treatments posed a significant difference in the outcomes among these units, that is the test units. So, a replication of treatment are allocated to experimental units at random. The design can, however, measure only one type of variance which occurs between the treatments. The only concern in this design is that the impact of all the extraneous variables, such as its price, advertisement, and competitor strategies, are assumed to be constant over all the treatment groups. It is this reason that this design is not very popular for use in marketing research problems. So the basic rule or the sequence of activities in this design will be: first is sampling. First of all, the sampling will be done. Then a random grouping. Third step is treatment, and then compare the results. This is the basic rule of completely randomized design. Let us understand it with the help of a conceptual diagram. Each experimental unit is randomly assigned to a random group to receive a different treatment. So please remember this basic rule. Like we have taken an experimental group that is whole subjects that is selected based on sampling. The first step in this design is sampling. So we have taken an experimental group on sampling. Then we have randomly assigned into group 1 and group 2. The second step is random groupings. So from the experimental units selected based on the sampling, we have done the groupings on a random basis into group 1 and group 2. That's the start. Third step is treatment. So we have applied treatment 1 and 2 on both the groups, treatment 1 on group 1 and treatment 2 on group 2. The fourth step is compare the results. Then after applying the treatments on both the groups, we have compared the results. This is the fourth step. Dear students, please learn this basic rule: sampling, random groupings, treatment, and compare results. Let us understand it with the help of an example. For example, a researcher wants to conduct an experiment to determine which environment is best suited for studying, a library in one's room or outside. एक रिसर्चर एक एक्सपेरिमेंट कंडक्ट करना चाहता है जिसमें कि वह ये देखना चाहता है कि स्टूडेंट्स के लिए कौन सा एनवायरनमेंट सबसे अच्छा है स्टडी करने के लिए, एक लाइब्रेरी या फिर उनका अपना रूम या फिर आउटसाइड। तो यहाँ पर यूनिवर्सिटी के 30 स्टूडेंट्स का एग्जाम्पल लिया गया है। We will follow this basic rule. We have taken a sample of 30 university students. This is the first step, that is sampling. Then second step is random grouping. So we have randomly assigned it into 3 groups. Group 1, 10 students. In group 2, there are 10 students, and in third group also 10 students. Then we have applied 3 treatments. Treatment 1, 2, and 3 on the groups. Now, the treatment 1 is library, means जो हमने 30 स्टूडेंट्स का ग्रुप लिया है उनको रैंडमली असाइन किए 3 ग्रुप्स में। हर एक ग्रुप में 10 स्टूडेंट्स है और हर एक ग्रुप को 3 ट्रीटमेंट्स दिए हमने। अब फर्स्ट ग्रुप को ट्रीटमेंट 1 दिया है कि वो स्टूडेंट्स हैं लाइब्रेरी में स्टडी करेंगे। सेकेंड ग्रुप को ट्रीटमेंट दिया है ट्रीटमेंट 2, मीन्स द स्टूडेंट्स विल स्टडी इन देयर ओन रूम। Third group has been given a treatment 3, means the students will study outside. So this is the third step. Fourth step is compare results. Now we will compare the results of 3 groups, that is treatment 1, after applying treatment 1, 2, and 3. Now we will compare the results. So this is the completely randomized design. Please learn its basic rules: sampling, random groupings, treatment, and compare results. There are 4 basic steps in this rule.